Welcome, colleagues, researchers, clinicians, educators, and students. This presentation is prepared by Brian Geniuson, research scientist. External environmental forces function as continuous biological inputs that shape human physiology across molecular, cellular, systemic, and organism-level domains. Light cycles regulate circadian timing through photic entrainment mechanisms centered in the suprachiasmatic nucleus, synchronizing endocrine rhythms, temporal gene expression, metabolic regulation, and whole-body physiological coordination. Ionizing radiation, including galactic cosmic rays and solar particle events, induces DNA damage, oxidative stress, and genome instability signaling, activating repair systems such as p53, ATM, and ATR pathways. Gravitational load variation alters mechanotransduction, fluid redistribution, musculoskeletal loading, cardiovascular regulation, and structural cellular integrity. Environmental electrophysiological factors may further influence membrane dynamics, signaling behavior, and adaptive cellular responses. Although this domain remains exploratory and requires continued empirical validation, these environmental inputs are processed through interconnected biological systems that operate as integrated regulatory networks. Circadian regulation governs temporal physiology and synchronizes internal biological rhythms with external environmental conditions. Neural adaptation reflects central nervous system plasticity in response to environmental variability, shaping cognition, perception, and functional responsiveness. Immune modulation involves cytokine signaling dynamics, inflammatory balance, and host defense recalibration under environmental stress. Cellular stress responses include oxidative stress accumulation, mitochondrial dysfunction, DNA repair activation, apoptosis, and autophagy. Metabolic regulation integrates energy homeostasis, endocrine signaling, substrate utilization, and adaptive bioenergetic balance across changing environmental contexts. The downstream biological output of these processes is expressed through physiological adaptation, system dysregulation, and long-term disease risk modulation. Physiological adaptation reflects successful homeostatic compensation and the maintenance of functional stability. System dysregulation emerges when compensatory capacity is exceeded, producing instability across circadian, immune, metabolic, and cellular domains. Over extended timeframes, these disruptions contribute to increased susceptibility to oncogenic transformation, cardiometabolic dysfunction, neurocognitive decline, and other environmentally influenced pathological outcomes. From an evidence perspective, human observational and clinical data support the biological impact of circadian disruption, radiation exposure, and altered gravitational conditions on physiological systems. Mechanistic and preclinical research further supports underlying pathways involving DNA damage, oxidative stress, inflammatory signaling, mitochondrial dysfunction, and adaptive cellular remodeling. The convergence of these evidence tiers reinforces the biological plausibility and translational relevance of environmental influence on human systems. Key scientific findings are as follows. External environmental forces represent biologically active determinants of physiological behavior rather than passive background conditions. Their effects propagate through interconnected regulatory networks rather than isolated pathways. The balance between physiological adaptation and system dysregulation determines resilience, vulnerability, and long-term health outcomes. A systems-level analytical approach is required to fully interpret the interaction between environmental exposure, biological response, and disease risk modulation. This body of work represents a structured research output designed to support scientific review, interdisciplinary collaboration, and advanced systems-level investigation. By organizing environmental influences into a coherent biological architecture, it provides a scalable platform for deeper exploration across radiation biology, chronobiology, environmental physiology, and space medicine. For researchers, clinicians, and institutions seeking expanded analysis, extended monographs, premium research materials, and advanced scientific frameworks are available through Natural Scientifics. These resources provide deeper literature integration, higher resolution mechanistic mapping, and comprehensive systems-level interpretation for continued research and translational application. This presentation was prepared by Brian Geniuson, research scientist. This content is provided for educational and scientific purposes only and is not intended as medical advice, diagnosis, or treatment.